I'm Mark Abbott. I'm the Managing Director of Peak Proteins, which is fully a part of Signature Discovery, which is an integrated contract research organisation. It probably started as an undergraduate, which was a long time ago, when I first actually started doing experiments, and I really got a kick out of doing experiments, not really knowing what the outcome was going to be, and have to struggle to solve problems. That wasn't really in protein and cell science, but that then led on to a PhD and a postdoc where I was working on membrane receptors and then a carbohydrate binding protein. And I just love the sort of research challenge of trying to understand proteins and solving problems with proteins. And then that led on to a whole career working with proteins, basically. Protein science is pretty much as, well, old as the hills. So Fred Sanger was awarded his Nobel Prize in 1958 for the crystal structure of insulin. And two of the most commonly used techniques that we still use today in peak proteins, so protein chromatography and SDS-PAGE, they've been around since I was a child or since before I was even born. So you sort of think, well, what has changed? What's new? So I think protein science is something that has constantly evolved such that what we do now is just a complete revolution, but it's not. It's very much been a revolution by evolution, if I can use that phrase. I mean, I guess proteins are just critical reagents within drug discovery. So proteins are regularly the targets for drug discovery, trying to find a new cure for cancer. You know, we'll often focus on the aberrant protein in a disease process. Similarly, over the last probably 30 years, then proteins have become sort of drugs within their own right. So the ability to make proteins as drugs at very large scales has become super important. Three things that have particularly changed over the last 20 years within protein science. So cryo-EM obviously has become super important and is now starting to really impact on protein science. Gene synthesis, which I guess was really starting to develop 20 years ago, now just underpins so much of what goes on within protein science. And then the new kid on the block really is AI, and the impact of AI on what we do is becoming really important. I guess the one thing that I would say though is that the bedrock of all of this is the ability to make high quality proteins. And that still relies upon the very traditional techniques. And I have an old colleague who used the phrase protein whisperer. And that's the person who has the ability to tame proteins and thereby make them in really high quality with really good stability. They're targets in drug discovery. So you've got a disease, be it inflammation, oncology, infection, that will frequently involve an aberrant protein. So being able to target that with either chemicals or biologics. So the ability to make that protein and then screen against that protein is absolutely critical. And then they are biologics in their own right. So, you know, starting off with the monoclonal antibodies, but more recently with all sorts of different protein modalities, which are drugs within their own right. When I started, recombinant DNA technology was really at its infancy. So drug screening was essentially, yes, it was biochemical to some degree, but even the biochemical screening just used crude tissues and things like radioligand binding assays. So Whereas nowadays, you know, you can order hundreds of variants of synthetic genes. You can run lots of parallel screens against a particular target. You can design exactly how to run that screen using the recombinant DNA technology. And of course, you can support all that with fantastically detailed, high resolution structural biology. So, I mean, it is just so different. And it all has been an evolution, but the end result is that revolution. I'm pretty sure AI is only at its infancy in terms of the impact that AI has on protein science and drug discovery. And I'm pretty sure that there are others who are better informed to comment on that. I'm just very aware that it's starting to impact quite significantly, certainly on what we do, and I'm sure on a lot of other companies. You know, quite a lot of our clients are well invested in AI. A second technology that I think is going to become increasingly important is cryo-EM, and I've already mentioned it, but I'm pretty sure that cryo-EM, the applicability of cryo-EM in drug discovery is going to increase. So the resolution of those structures will increase. I suspect the ability to go through that whole process faster and faster will also increase. The one comment I'd make is that I think the bedrock of doing all that will still remain the ability to produce really high quality proteins by those people who I call protein whisperers. So at any one time, we always have probably 40 to 50 different projects. So some of those will actually be quite straightforward, and we can just sort of churn those through and complete them. But within that 40 to 50 projects, there are always perhaps 20 or so that are tricky, that you try and anticipate what's going to happen, but you do the first experiment and the best laid plans completely fall apart. And we love that sort of project where you do something, it doesn't go to plan, and you actually have to sort of really look at the data, look at your literature, talk to the client, and try and sort of solve those problems. And that sort of project is always sort of coming through peak protein stroke signature. And, you know, that's what we love doing. We love solving those sorts of problems. Even though sometimes, you know, there'll be problems where we have to hold our hands up and say, actually, we can't progress this. But it's super, super satisfying when we are able to solve those problems. And I guess it's that sort of almost goes back to where I started. You know, being able to sort of solve those problems is great, and still gives me a big kick.
Yes,